Namaste students, I'm JB Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I'm going to discuss about the topic downstream processing step number 3. So in this video I'm going to discuss about the product purification. So first I need to discuss about the downstream processing. Downstream processing is the processing which is conducted after the fermentation process, and there are various types of downstream processing which include the removal of the insoluble, product isolation, product purification, product polishing, and then they are sent to the market. So I have discussed about the removal of the insolubles, removal of the and product isolations. So in this video I am going to discuss about the product purification. Product purification is done to separate those those contaminants which resembles the product, which are similar to our product, and they are contaminant. They have some properties which are similar to our product. Either they are physically similar to the product or they are chemically similar to the product. So uh, these compounds, these contaminants must be removed so that our product can be purified. This step is very expensive. The product purification is very expensive and it requires very sensitive and some sophisticated equipments. The significant fraction of the entire downstream processing is very hard. The examples of operation include affinity chromatography, size exclusion chromatography, reverse phase chromatography, uh, crystallization, and fractional precipitation. So these all are the examples. In this lecture, the chromatography will be discussed. The chromatography is a technique which is used for the separation of the mixtures. The mixture is separated on the basis of either color or any other basis in which a mobile phase will be present. Okay, the passive mixture are dissolved in the mobile phase, and there will be a stationary phase on which our samples is loaded. Okay, so samples are passed in mobile phase through a stationary phase, which separates the sample uh, to be measured from one molecule in the mixer and allows it to be isolated. So sample will be separated in the chromatography techniques, and various chromatography methods are there. First is the ion exchange chromatography. What is ion exchange chromatography? Ion exchange chromatography, in this we use charge stationary fields to separate charged compounds. The reagents that carries charged functional groups which interact with oppositely charged groups for the compound to be retained. So the charged groups are separated. So suppose that there is a positive charge and there is a negative charge of the our molecule is, suppose it is negatively charged. So for negatively charged sample, we are adding a stationary phase of the positively charged. So our sample in the mobile phase will bind to the positively charged stationary phase, and it will be bind to each other. This will lead to the separation of that particular compound. The positively charged protein bind to the negatively charged protein. So in this ion exchange chromatography, we are using some beads which are given a charge. So these beads are stationary phase. If we have a positively charged beads, so negatively charged samples such as proteins will bind to that and they will be separated. For negatively charged protein, we are using these beads, and the negatively charged beads will float so the column. So they will be separated by this method. So what is the basic things in an Excel chromatography? Ion is an atom or molecule that has lost or gained one or more valence electrons. When if lost, then it is given as a positive or negative charge. If it lose its valency, then it get any kind of charge like positive charge and negative charge. And anions are the negatively charged ions formed. Anions are negatively charged ions formed when an atom gains electron in a reaction. And anions are negatively charged because there are more electrons associated with them than there are protons in the nuclei. So they have more, more electrons. So they are not considered as a negatively charged. Cations are positively charged formed when an atom loses electrons, and it means that they have less electrons than the nuclei. So they are positively charged. And by this, they will get any of the charge by losing. Now, affinity chromatography. Affinity means we have different. We know that what is the affinity? Affinity is the binding capability of one molecule to another molecule. Means affinity is the strongness between the binding capacity of two molecules, such as the antigen and antibody reactions. If antigen and antibody are they are complement to each other, then they will follow the clock and key model, which will allow the antigen and antibody to bind to each other very strongly. If they are very specific to each other, they will bind very strongly. If they are not specific to each other, they are low. So in this affinity chromatography, molecules are separated on the basis of their affinity. So affinity chromatography separates the protein of the interest on the basis of their reversible interaction between the and its antibody coupled with the chromatography beads. So protein is at the act as an antigen and antibody will bind to it. With high sensitivity and high resolution and high capacity of the protein of the interest, purification level in the order of the several thousands fold are achievable. The protein of interest is collection in a purified concentrated form. The biological interaction between the antigen and protein of interest can result in the electrostatic reaction. Some van der Waals interaction can be formed, some hydrogen bonding can be formed. You the protein of interest from the affinity beads, the interaction can be reversed by changing the pH. So if you want to change the interaction between the molecule, we can change the pH so that the interaction can be changed. Some another bonding can be made between the molecules so that the interaction can be changed. The interaction can be higher. The interaction can be strong. The concentration effect enables large volumes to be processed. The protein interest can be verified from high level of contaminating substances. Making antibodies to the protein of interest is expensive, so affinity chromatography is the least economic choice of the production of the chromatograph. At the last, I will explain it with the diagram and the size exclusion chromatography. So, what is the size exclusion chromatography? Size exclusion chromatography is the chromatography method which use the size of the molecule for their separation. The large particles are separated from the small particles. It is also known as gel permeation filtration chromatography. It separates molecules according to their size and low resolution pollution. The example of size exclusion chromatography chromatography is uh, gel electrophoresis. We separate the particles on the basis of their size, and gel electrophoresis, in which grooves is a stationary phase, and some buffers look like a mobile phase, and they are separated. They move on the basis of their charged particles, and they are separated on the basis of their size, because larger molecules will not be passed through the agarose, and small molecules will easily pass through the agarose, and travel in they will diffuse in the agarose very easily, but large molecules will not diffuse in the agarose very easily, so they will be easily separated. So large molecules, for the separation of the large molecules in the material, they can be easily separated. Now liquid chromatography. Liquid chromatography in this mobile phase is liquid, and it is carried out through a column on a plane. The example is the HPLC, high performance liquid chromatography, or liquid chromatography. In the HPLC technique, the sample is forced through a column that is packed with irregularly or spherically shaped particle, or a porous monolithic layer. Means it is a stationary phase. The liquid is flown in liquid chromatography at higher pressure, which allow the molecules to pass through, and the monolithic layer will separate all these particles. So this is the basic principle of the liquid chromatography. And next, the crystallization. Crystallization is a process of formation of solid crystal by precipitating form a solution. They are melted and they are deposited directly from a gas, so that they can make some crystals. The chemical solid liquid separation technique, in which mass transfer of solid form and the liquid solution, they pure solid crystalline phase also occur. So this is the method. So this is all about the crystallization method. Uh, so affinity chromatography and size exclusion chromatography, I must explain it with the diagram. This is the affinity chromatography diagram, where the antigens are deposited with the antibody. This is the antibody specific for the antigen, and these antigen will bind to this antibody molecule, and they will be separated. This is the antibody of different specificity. So in a mixture, we are adding different kinds of antibodies. The specific antigen and antibody interaction will be conducted. So suppose that there are three antigens. So for those three antigens, we are adding three different types of antibodies. The matching antigen antibody will bind to each other, and the remaining material will be separated. This is the diagram of the size exclusion chromatography, in which the molecules with larger size will remain in this portion, and the molecules which are smaller in size will pass through the surface, through the gel or whatever the column is there. So they will pass through the column very easily, but the larger particles cannot pass through the column because of their larger size. So I hope that it is well understood to all the students. So this is all about the Downstream processing step number 3. I hope it will be understood by the students. Uh, the students can be understand about uh, the third step of the downstream processing, that is the product evaluation, which is very typical method. And in this, I explained about the chromatography methods, various chromatography methods, including the size exclusion chromatography, various chromatography methods like ion exchange chromatography, size exclusion chromatography, affinity chromatography, liquid chromatography, and crystallization. So these were explained. So questions which can be asked in this lecture. Uh, Explain the third stage of the downstream processing. What are the processes which are involved in the product purification? So I hope that student can explain it. I have uploaded the teaching notes on the portal. So my lecture is over here. Thank you very much.